As we know, microbes are found everywhere, from the environment to some foods we consume and even on us. The human body is home to trillions of microbial cells. They have evolved to live on and within, assisting in functions such as digestion, immune homeostasis, and more recently discovered, brain health. There are an estimated 100 trillion microbial cells that call us home. When we compare that with our human cells, of which we have around 30 trillion, it's easy to see that there is an abundance of our native microbes outnumbering human cells. These microbes inhibit our skin, mouth, and nasal passage, but the vast majority of them are found in our gastrointestinal tract, or our guts. With over 2,000 species of microbes, including bacteria, archaea, fungi, and eukaryotes, these are collectively known as our microbiota. Similar to fingerprints, no two people share an identical gut microbiota. The balance of microbiota in our guts has shown to have an effect on our health and well-being. This presentation aims to provide an overview of the human microbiota and what effects these microbes have on our bodies. In 1683, Antony van Leeuwenhoek first observed bacteria from cultures he obtained in his mouth. In 1854, Filippo Puccini discovered bacillus in the human gut after investigating the cause of the cholera epidemic in Florence, which he named Vibrio. He based his studies on the concept that there was a causative agent making a large population ill with a high mortality rate. He concluded that Vibrio was in fact the specific cause of cholera, though his findings were not accepted by the scientific community. Robert Koch formulated the same theory and was later awarded a Nobel Prize for his work. Bacteria in the body has been heavily influenced by these early works, and we tended to regard bacteria as only pathogenic. There was a belief that not all were pathogenic, and some bacteria actually benefit the body in the form of probiotics. It is only recently that these good bacteria have been studied more in depth. With modern technologies such as 16S ribosomal RNA gene sequencing, scientists are now able to understand the key of our microbiota in normal biological processes as well as disease. Of the millions of species of bacteria, only an estimated 1,400 are known to be pathogenic. Modern microbiota research is as much interested in the effects on the health as well as being on disease. Our gut has a symbiotic relationship with our body. We provide a nutrient-rich environment in which the microbiota live, and the microbiota in turn aids in tasks such as digestion, nutrient scavenging, and more recently discovered brain health via the brain-gut axis. If there was a shift in this symbiotic relationship, whether it be pathogenic invaders or prescribed antibiotics, our microbiome could become dysbiotic, and this is where things go wrong. Some studies suggest that bacterial infections can lead to molecular mimicry, where a foreign or non-self antigen shares the same structure as a self-antigen. This molecular mimicry causes the expression of two T-cell receptors on one T-cell. The host is left exposed as the T-cell is able to react to both self and non-self antigens, and this is where it can potentially lead to autoimmune diseases. The composition of the gut microbiota is dominated by the phyla Firmicutes, Actinobacteria, Proteobacteria, and Bacteroidetes. It has been found that those individuals with a higher animal intake have higher levels of bacteroids and ruminococcus in their guts. And Provotella is generally found in individuals with higher plant-based diets. Butyrate-producing bacteria such as Clostridium, Roseburia species, and lactic acid bacteria like Lactobacillus have shown to have benefits in anti-inflammatory and pathogen-excluding properties. Cross-feeding on metabolites between members of the microbiota can also be important in the maintenance of microbiota homeostasis. For example, lactic acid bacteria and butyrate-producing bacteria can form an important consortia, with the butyrate-producing bacteria able to feed on the lactate. So how do we get our microbiota, and why are we all different? It all starts at birth. When in the womb, a neonate's gut is thought to be almost sterile, with low numbers of microbiota. Once the neonate is passed through the mother's vagina, it will become colonized with her vaginal microbiota, such as Lactobacillus species. However, those neonates that are delivered via caesarean section have shown to lack these microbes. Instead, they display higher levels of skin bacteria like Staphylococcus epidermidis. In 2017, a group of researchers studied the difference between the gut microbiota of infants delivered vaginally and those that were delivered via caesarean section. They found that after 5 days, there was increased numbers of bacteroidetes in the caesarean section subjects when compared with the vaginal delivered babies. So from this study, it seems that in infants' microbial makeup is heavily influenced by the maternal microbiota. Though this is not the only influential factor on how our microbiota is formed. Within 6 months of development, an infant's gut will go through many changes. Some researchers believe that whether an infant is breastfed or formula-fed influence the formation of the gut microbiome. In addition to this, environmental and physiological factors can also affect the types of microbes that are more abundant than others. Factors such as diet, whether they be vegetarian or high-fat diets more commonly seen in the Western world. The use of antibiotics can also disrupt these, as they kill off the good vital bacteria, leaving the pathogenic ones to survive and thrive. So why do we need our gut microbiota? The gut microbiota is now thought to be an essential organ of the body that works by aiding the host's body by synthesizing nutrients from intake of food, detoxifying, enabling signals to enhance epithelial renewal, excluding pathogenic bacteria, and producing antimicrobial products, and finally, the development of our intestinal immune system. Vitamins such as vitamin K cannot be made in the body. It is a byproduct of our microbes. So if the body lacked gut microbiota, it would be unable to complete these vital functions, potentially causing disease and other health problems. What direction are we taking to learn about the microbiota? To try and piece the puzzle of the relationship between the gut microbiota, gut health, and how it interacts with the body, the Human Microbiome Project was established in 2007 by the United States National Institute of Health, commonly known as the NIH. This research cohort went in with a mission to gain an in-depth understanding of how the human microbiome affects the body. They also aim to understand whether individuals share specific microbiota or whether they are all different. And they also wanted to establish whether changes in our microbiota are correlated with any other health changes. In phase 1 of this 10-year study, the Human Microbiome Project used 300 subjects, both male and female, aged 18 to 40. Before the subjects were able to participate in this study, they were extensively tested to ensure that they were free from disease. They were also unable to take prescribed antibiotics, nor were they allowed to take any prebiotics or probiotics during this time, as this could potentially affect their microbiota. Using metagenomic analysis, researchers were able to map out each of the individual's microbiome. The second phase of the study was mapped out into 3 separate areas. These included birth and pregnancy, whether there were significant differences in the microbiota due to delivery route, and whether we share the majority of our microbiota with our mothers. Gut microbiome and autoimmune disease. Are we all the same, and can we conclude whether our gut microbes cause autoimmune disease or whether they enhance something that is already there? And finally, type 2 diabetes. Concluding in 2017, the Human Gut Microbiome Project concluded that there was in fact a correlation between the human gut microbiome and health and disease. Though there is still much more to be studied to be able to learn whether the balance of microbes we display, species and abundance, is a cause or effect of disease. This project, however, has paved the way for new research with new ideas and approaches, and much broader studies can be conducted. So what do we know about our gut microbiota and how they assist in regulating our bodies? Firstly, they play a major role in digestion and nutrition absorption. Without the presence of our gut microbiota, our bodies would have a difficult time digesting certain foods. Our guts are quite capable of breaking down simple sugars such as glucose or lactose. However, more complex molecules such as complex carbohydrate polymers, lipids, and proteins are a little more difficult. These microbes use the complex molecules as an energy source. By breaking down these, they produce byproducts called short-chain fatty acids. These short-chain fatty acids are a major source of energy for our intestines. Using butyrate as an example, this has an anti-inflammatory effect on our colon. If there was a shift in the production of butyrate, most likely caused by dysbacteriosis in our guts, it could lead to diseases such as inflammatory bowel disease and Crohn's disease, which is now believed to be an autoimmune disease. Shifts in our short-chain fatty acids could also have an impact on our obesity and nutrition. In 2016, the World Health Organization estimated that 1.9 billion adults and 41 million children are overweight or obese. And the Australian Bureau of Statistics estimated that in 2015, 2.5 million Australian adults were overweight or obese, with 25% of our Aussie kids. These numbers don't sound too alarming. Just think, these numbers have tripled over the last 50 years. Why is this important? Because numbers show that the world's weight is growing, and so will the prevalence of any preventable diseases that come along with it. If we start giving our guts the nutrients they need, this could assist our microbes in doing a better job in reducing weight-related illness. As previously mentioned, the gut microbiota also play an important role in the development and proper maturation of our immune system, helping the body recognize and mount appropriate immune responses. When there is an inappropriate response to microbes, the immune system can react incorrectly, and this has been linked to autoimmune disease. The autoimmune disease type 1 diabetes affects an estimated 1.2 million Australians, with 40,000 new cases each year. This is caused by the dysfunction of an immune response that causes the destruction of insulin-producing cells, where there is a TC-CD4 mediated attack on the beta pancreatic cells. Studies have found that the microbiota in the gastrointestinal tract could increase the onset of these CD4 attacks. In one study, young non-diabetic mice were infected with Siderobacter rodentium via oral gavage. These species showed to have high intestinal permeability, and lymphocytic filtration on the pancreas was increased. It was also found there was a slight decrease in microvilli. Children at risk of developing type 1 diabetes showed a high concentration between intestinal permeability and the alterations of their gut microbiota. When these studies were compared with healthy controls, they displayed to have high levels of certain species in their guts. A successive study also showed that the gastrointestinal microbiota of some children, ages 3 months to 3 years, had elevated species in their guts. The link between gut microbiota and type 1 diabetes can clearly be seen here, as the changes in the microbiota were found in those that displayed an autoimmune disease. If we have a quick look at allergies, which are also considered an autoimmune disease, they have become extremely prevalent in recent years. From almost non-existent 30 years ago, Australia is now the #1 country for allergy sufferers. There has also been an increase in food allergies. Though there's little study done, it can be seen that there is a clear correlation between obesity and autoimmune disease. This could all tie into our gut microbiota and whether they are happy and working correctly. And finally, recent studies have shown a clear correlation between our gut and our nervous system, known as the microbiota brain-gut axis. This is a quickly emerging area of microbiota research. The brain contains neurons that allow your brain to tell your body what to do and how to function, with the relationship between the enteric and the central nervous systems. The enteric nervous system is found in our gastrointestinal tract, and it controls our gut. The enteric nervous system is connected to the central nervous system via the vagus nerve, and the vagus nerve controls organs such as our gastrointestinal tract and our heart. It works by carrying messages to the brain and enables the body to function correctly. Researchers in 2014 were able to demonstrate, using mice models, that stresses have had an impact on our microbiota population. The researchers demonstrated that mice that were exposed to stress states had significantly lower amounts of lactobacillus species compared to the controls. So a shift in mood is a shift in our microbial population. It should also be added that serotonin, the neurotransmitter for happy feelings, is actually produced in our gut. In 2018, it's an estimated that 3.2 million Australians were diagnosed with an anxiety-related condition, an increase in previous years. Also, 1 in 10 were diagnosed with depression, another increase in recent years. So it seems that our gut affects many things that happen to us. What we need to distinguish is it the microbiota that is causing all of these issues, or are they enhancing something that is already there? It is hoped that furthering the microbiota research and how they actually interact with us, whether they benefit or they accentuate an underlying disease. We should also look at alternative medicines aiming for the reduction of antibiotics. Perhaps we could prescribe toxin blockers to block a specific toxin for a specific microbe and not to kill our microbiota altogether. Or maybe when we get prescribed antidepressants, should the doctor be looking at our diet? While we wait for more advances on this emerging topic, we should be asking, if we are a meta-organism formed by the aggregation of individual organisms, how do we assist them all in maintaining healthy functions?